It says going live in 1, but nothing's happening. So sit tight. Oh, shoot. All right. It did this to me yesterday too, everybody. It's saying I'm out of minutes. Rashi, can you try hitting go live? It won't let me go live. There we go. Thank you.
Is it still sharing your screen?
It should be. Yep.
Yeah.
Yay. Okay, we are live. Hi, everybody. Welcome to our AP Bio cram session. We are on night 3. I'm going to just wait a few minutes as people come through, give everyone a minute to get situated. I'm Kari Parnan. We'll do official introductions here in just a minute. So we'll give everybody about 60 seconds to get situated before we really begin. But we're so glad you're here. And while we wait, on internet. There we go. I was trying to get the music going. There we go. There we go. Yes, here's your favorite song, everyone. We're going to do this for about 30 more seconds as we wait for the rest of our group to get here. All right, that's it. All right, welcome everybody. We are here for our 2023 Fiveable Crams. I've got some amazing student hosts here with me. Let's get going. If you haven't already, make sure you're following Fiveable on social media. They post really fun videos. They post reminders. They post encouraging words for you to kind of lift you up and get you ready for the busy exam schedule that you're going to be facing this week. So I'd love to introduce our student hosts for tonight. And so in order to do that, they're actually just going to hop on and say a quick intro for you. So whoever would like to start first, go ahead.
I can go first. Hi, everyone, again. I'm Madison, and I am a student host coming to you guys from Illinois. It's a pleasure to be helping you guys again for the 3rd cram session.
Yeah, I can go next. Hi, I'm Rashi. I am a college freshman from Tampa, Florida, and it is super nice to see you guys again.
Hey guys, I'm Sahitha. I'm a high school senior from Dallas, Texas. It's good to be with you guys again.
All right, that was everybody, right? Everybody's good? All right, so I'm so excited that you're all here. It's Sunday night. Where I am, it's Michigan, so we are 7 p.m. my time. We're going to be together tonight for 3 hours, and I just want to encourage you to sit tight, to use the chat, to be encouraging, to be positive, and to be helpful, and then also use the Q&A on the right as well if you have specific questions. So tonight is our 3rd night. We're really going to be focusing on skills and drills and just overall test taking strategies. And then a lot of our stuff will be focused around science practices 1 through 3. And I'll get through that. I'll explain that a little bit more here as we go. And then tomorrow night, Mrs. Shoemate is going to be doing skills and drills, which will be covering calculations, graphing, and drawing conclusions. So I know a lot of you are feeling like, oh, when are we going to get to Hardy-Weinberg? And when are we going to get to chi-square? That'll be tomorrow night with Mrs. Shoemate. And so just sit tight for that. Obviously, you can review that on your own and maybe come with questions, but you will be covering that. And then on May 9th is our grand finale, which you'll get a whole review again of units 1 through 8, some test taking strategies, and then other tips and tricks. So we have a lot more time together, and I just am so excited that you're here. We've got over 200 students from across the country. Our overall agenda for tonight, if you can see there, welcome and FRQ strategies. Then we're going to overview the 3 science practices and then dig into each one individually. And within that, there's going to be tons of FRQ practice. So if you're looking for some more practice, I know the last 2 nights were really content driven, right? We went through everything. We went through it. So tonight we're really going to dig into, let's see what a test question looks like. This is something, if you've been to other cram finales, these are how to make the most of them. So first thing, take notes. You should have a piece of paper out. I even have paper here too, right? Like I'm hosting this, but I still have a piece of paper out because if you say something that I need to write down to think of for later, I've got something to write on. Also the Cram Hub. So if you look at the bottom of your screen in Crowdcast, or at bottom of your screen, there should be a greenish blue button that says AP Bio Cram Hub. And so that's where you're going to get slides, review packets, and replay. So if you missed the last 2 nights, you could get a replay as well. The group chat, like I mentioned, is for sharing tips and tricks, to participate when we ask questions, and then just to be kind and helpful. If the chat gets a little unruly or negative, we will just, we'll give you a warning at first, but then we'll just kind of take you off the chat, and you can still attend the session, but we would just take you off the chat. The Q&A tab is super helpful. It's for asking any questions. And then you can actually vote on the questions. There's an upward arrow and you can say like, yeah, I have that same question too. And so if you vote for those questions, we can kind of take a look at them. And if we have time at the end of tonight, we will do our best to answer those. I know last night, if you attended, we went straight to 10 o'clock. Like there was not a second to spare. And but hopefully tonight I tried to build tonight a little less intense so that we had time at the end to circle back and really get your questions answered. Some community agreements. Your teacher might do these in class where it's like class norms or a social contract, but it's basically just who we are as a community and what we're going to commit to for these next 3 hours. So be supportive and lift each other up. I saw a lot of that yesterday. It was really impressive in the chat where someone had a question and our teacher hosts were there to answer as well. But there were also other students who were like, we got this, we can do this, and helping each other out. Be patient. We still have 3 nights together, right? We have tonight, tomorrow, and the other and Tuesday night. So be patient, right? Learning this topic is hard. Thinking about this topic again in preparation for an AP exam is stressful. So just be patient. And if there's something that like you're really eager to go over, you can ask that question in the Q&A. And our teacher hosts are really good at answering your questions as well. And then finally, be kind, because we are all real people. I am live from my house. I'm in the basement. My husband's watching the 3 kids upstairs. So hopefully it's as quiet as it can be. But again, be kind. We're all real people, and we'll treat you the exact same way. And now there's also a Cram Experience winning that you can have. So you can win $100 by sharing your Cram experience. So if you post about your finale experience on TikTok or Instagram, you can use the hashtag #5ablecrams and tag Think5able. So those are the things you have to do in order to get seen. And make sure you're also following Think5able. So you can make a little post about what it was like in the crams, or you can make a post about how the crams helped you. And it says you can win by making us laugh out loud or getting a ton of views. And so several students will win each week, and you can win $100, which is pretty amazing. So I hope you win. I hope a biology student wins. That would make me really happy. So yeah, that's enough of that. Officially welcoming you. I see our attendance numbers are still going up. So I know that students are, it's not quite 7 on the dot. I'm rarely ever on time to things, so I get it. So as our attendance still continues to rise and people enter, I'd love for you to just in the chat give a word of encouragement to everyone who's here right now. So students of mine would say that I'd ask them for like an affirmation, like what's something kind you can say to the general group of students, or maybe it's like, what's your motto, or what's something that helps you grab that confidence. So go ahead and share that in the chat. I'd love to see what you say. So share in the chat. I love it. We got this. Yes, this test does not determine you. Yes, 100%. Oh, you're all so sweet. Got this. I love it. We're all future 5 scores. Cool. You're all so great. Yes. I love what Jasmine said. Even if you don't feel like you're going to do well, it's not the end of the world. It's just an AP test. And, the fact that you are all here, you are showing up. And so that says something about you as a person, as a student, and that you believe in yourself and that you're going to put in the work. And because you're putting in the work, I know you're going to do well. So I'm really, really glad. Good job, everybody. All right, I just want to jump into kind of sidestep into some things about the day of the exam. I know on Tuesday night we'll really hit these hard because it's the night before the exam, but just into anticipation because, you might not have some of these things. You might not realize you need them. So pencil and pen. Oh, this is for tonight's session. Sorry, I thought I was going ahead. So tonight, yes, you need a pencil and pen. You don't need a calculator. That's my bad. Blank paper, water, and snacks. Yes, all of those things. I thought I was on my slide for what do you need on the day of. All right, so I'm going to make a quick poll so we can get some practice using the polls. So if you slide over to where there's like a little graph on the right, there are 3 options. So what is better? Sleeping in, going to bed early, or taking naps? I love this question. Oh, the poll is visible, right? You can all see this. Ooh, it's like 50-50 between sleeping in and going to bed early. I'm really surprised. And see, taking naps. Y'all don't take naps? Gosh, I'm telling you, naps are amazing. Like if you can get a 20-minute nap in when you're feeling exhausted, there's nothing like it. Awesome. Thank you, everybody, for participating. So that's just a question, thinking about it. Sleep is important. So whether you go to bed early, you sleep in, or you take naps, make sure you're taking care of yourself this week. I know a lot of you probably took AP exams last week, and you probably have more than one this week. So give yourself sleep. I know one of the best pieces of advice somebody gave me in college, I was preparing for a calculus and an organic chemistry final that they were going to be on the same day. And I was about to pull an all-nighter and stay up all night studying. And someone said, if at this point you don't know it, there's nothing you can do overnight that's going to make things better. The best thing you can do is go to bed. And they were absolutely right. I completely aced my calc exam. I did okay on orgo, but organic chemistry wasn't my thing. But it was just the best advice. I was like, you know what? Yeah, I could drink coffee all night and I could stay up all night, but how would my brain be working that next day during both finals? And so the best thing for me was to just go to sleep. So I encourage you this week, get yourself some sleep and take care of yourself. We went over this on nights 1 and 2, just some other logistical things, right? The exam is Wednesday. It's at noon local time. It's covering the 8 units below. And it's okay if your teacher didn't get to every unit. I saw some students talking about that yesterday, like, oh my gosh, we didn't even do ecology. Your teachers are doing the best that they can. And even if you didn't get to it, there's so much, especially ecology. There's so much on the exam that's all just reading diagrams, reading information, and applying it using just your science skills. So regardless of if you've learned every little detail, you still have a really good chance of being successful. The exam is 3 hours. So these 3-hour cram sessions are really good because we are kind of modeling that, like, hey, your brain is going to be working for 3 hours straight. And so we're modeling that for you in the last few days that we're together. I'm not sure if you can see this picture well, but it basically shows the exam layout. And you might be like, my teacher's already told me this 100 times. Like, why are we still talking about it? Even if there's one student who hasn't heard this, it's worthwhile to know. Like, it's helpful when you get to AP exam day that you are walking in knowing I have to answer 60 multiple choice questions, or I should say, I get to answer, right? I get to answer 60 multiple choice, and I get to answer 6 FRQs. They're worth 50% each, right? So if you're stronger in one than the other, totally good. A good portion of the multiple choice and FRQs do cover our science practices that we're going to get to tonight as well. I want to start with just some FRQ strategies because we're going to jump into some practice here in a minute. Has anyone ever heard of the ATP or KISS method? Go ahead and write in the chat if you have. If your teacher has ever said ATP, or if they've said KISS for a strategy. You haven't? Okay, good. Yes, one of you has. Okay, so ATP means answer the prompt. And I say keep it simple, silly, actually, instead of keep it simple, stupid. But yes, keep it simple. ATP means answer the prompt. So the prompt is going to ask you to do something, and all you have to do is answer it, right? And keep things simple. So if the prompt is really intense and it's like self-signaling and it's got all these abbreviations and all these enzymes and all these cascades happening, but the prompt just says, identify the location of such and such thing, the diagram might even have the word cytoplasm on it, right? And it's wanting you to just find it and answer it, right? So answer the prompt. These are some tips from Mrs. Shoemate that she'll share tomorrow too, but I love them, so I took them for tonight. Imagine the AP test is like an Easter egg hunt. So you're like hunting for answers because the answers are generally there, which is amazing. Graphics usually have a lot of answers. Yes, exactly. Answer the question before looking at choices. This is one of my favorite strategies for multiple choice questions because sometimes the multiple choice question answer options are so long and you're like, wait, A and B sound almost the same. So it's helpful to cover up your answer options and try to just answer the problem on your own. Okay. The KISS is keep it simple, silly. Yeah. So don't, if the problem's about cell signaling, don't go talking about how you know about cell signaling so well and defining every single word when all they're wanting to know is, what's happening during this one part of the diagram. So just keep it really simple. Awesome. Really nice. Everyone's feedback and ideas are great. So in summary, ATP means answer the prompt. Number 2, KISS. Keep it simple, silly. Don't go wild and putting a bunch of stuff on the paper because you think it's going to give you more points because you said more. That's not true. The AP reader will be really annoyed and it will be harder for them to figure out what you're trying to say. All right. Good. Keep it simple. All right. And another way to keep it simple, silly, and to just answer the prompt is know the task verbs. This is just a handful of them, but these are the most common. So when you see them in bold, that's exactly what they're asking you to do. So describe. It'll say describe in bold, and then it'll say words after that. So describe is what's happening. So you just have to write a simple sentence. Explain, it's why or how something's happening. It's a simple sentence. Identify is just a one-word thing, right? It can say DNA is found in the nucleus in a eukaryotic cell. Justify, you're going to want to prove something. Yes, I see someone writing in the chat that circling the verbs helps, right? Like if you've ever taken, especially like in physics, I feel like when I taught physics, I always made my students highlight and circle and box and be like, hey, this is information that's important. So absolutely circle things, underline things, annotate every question, write all over it because that is a huge test taking strategy. Analyze, so you're looking at something, describe what's happening, you're providing your own insight to it. Predict, I agree, predict is difficult. Yes, you paid for the test, you can write on it, absolutely. Predictions are hard for people because you feel so unsure, like who am I to predict this? I'm not the scientist behind this, but with prediction, just use your best science logic and say, and like be confident in, okay, well, if X and Y are showing this kind of relationship, I can predict that it will continue to do so. Or predict, a really big one for predict that I've seen is when you have like an enzyme that is no longer in an environment that's ideal. So pH changed or temperature changed, or something like that, right? So the enzyme, if the environment changes, what's likely to happen? Predict what's going to happen, right? So if the environment is changing, the structure might, good. Yeah, I see people saying the natures, the structure will change and therefore its function will be hurt, right? You can keep things really simple with your responses. So give it your best prediction. You might as well try. Usually you want to follow your prediction with evidence. Yep. Good. Awesome. Okay, so here's practice. I'm going to give you just a few minutes to read this prompt and just jot down some ideas. If you can't see this very well, someone yesterday said that you can zoom in on your screen to see it a little bit better. And I want to, before I hit the timer, I want to address something I'm seeing in the chat. If you were to answer something and say, oh, because it works like a lock and key, they can't give you points for stuff like that. Like if you say, oh, the mitochondria is the powerhouse of the cell, you can't do that. So I think yesterday somebody said you could zoom in, like if you hit Control plus. I'm not sure if that works on your end. Okay, I think you can zoom in maybe. Oh, someone's on a phone. Oh, brother. So if you're on a phone, you should be able to have access to the slides, but I'll just read it for everyone. How about that? Okay, people are doing full screen. Read the chat. People are explaining how to do full screen. Oh, I see it. Yep. So the slides, if you go to the green button at the bottom, it says AP Bio Cram Hub. Oh, it's not in the hub? Oh, I'll just share the slides then. Hold on. Hold on. All right. I'm going to put the slides into the chat unless someone has already done it. Not working? Okay. So there are the slides. Hopefully they work now. We are on slide number 16. Yeah. Okay. I'm going to go ahead and hit the timer and give you a few minutes to just look at those task verbs, think about what you would say. There we go. All right, I'm seeing some good responses in the chat. I'm going to go ahead and move to our scoring guideline. Now, I see a lot of you saying, oh, Unit 6, like, I don't remember this, or it's really hard. But again, when they've given you a visual representation to start with, and things are labeled for you, right? Like if I go back to my slide, they've labeled for you here that we've got DNA, and then primary transcripts, and then mature RNA, and then protein, right? So the very first question is talking about how the primary transcript is 15 kilobases long, but the mature RNA is only 7, and it's wanting you to describe the modification that most likely resulted in the 8 kilobase difference on the mature RNA molecule. So first, describe. So A, I'd even write the letter A and write you could either talk about removal of introns specifically. You could say, the removal of introns from primary transcript to mature RNA resulted in that 8 kilobase difference. Or you could have just said RNA processing. This is where I get really excited about FRQs, because sometimes there's more than one way you can say something and still get the point. Okay, so if you're like, I don't know what I'm going to say, or what if I say it wrong? They are right. The AP readers want you to get the points, right? They really do. They're not sitting there, putting red ink everywhere and wanting you to fail. They are trying to find ways to give you the point. And so especially when I see that on a rubric where someone could have said specifically removal of introns and someone else mentions RNA processing, as long as you connect it to what is being seen in the diagram, you'll get your point. The next word, identification of where this is happening. The diagram also kind of gives you that too, right? Like you can tell that there's this outer membrane and then this inner membrane, and that's where the DNA is. So hopefully some light bulbs go off. And then I'm pretty sure the prompt also mentions that it's a eukaryotic cell. Yeah, it says it's gene expression of a eukaryotic cell. So already they're telling you it's eukaryotic. And so you should know, identify, where does this happen? Where does this change happen? It's happening in the nucleus. Generally speaking, for identify questions, you could technically just write the nucleus, but to be safer, you should just say this process is happening in the nucleus. Okay. For B, it's wanting you to predict the length. And so you could specifically say the length will be 15 kilobases long, or you could say it's going to be longer than the mature mRNA. I always say be more specific. If you said splicing, you probably would get the point because that is RNA processing. Yeah. And then your justification for why that would be a longer mRNA, mature mRNA sequence is because mRNA processing does not occur in prokaryotic cells. Yeah, really nice. Okay. They cannot give you half points. So this is really good. This is why it's nice. It's either a 1 or a 0. And also, I'll talk about this in a little bit, that you can't contradict yourself. So if your justification said, in prokaryotic cells, they add a poly A tail or a GCAP or something like that. Yeah. So RNA processing typically doesn't occur in prokaryotic cells because eukaryotic cells, first of all, their RNA, mature RNA, has to travel through the cytoplasm, right? And so there are a lot of things that could potentially degrade that message. But they also have translation happening at the exact same time. So the need for cutting out your introns and splicing your exons back together isn't quite necessary. Someone's other question is, why is it 15 kilobases? So if this was expressed in prokaryotic cells, the introns wouldn't be taken out. It would just be expressed as is. Because remember, in a prokaryotic cell, like a bacteria, everything is happening out in the cytoplasm. So as soon as transcription is happening, translation is happening as well. And so there isn't like time for RNA processing to happen. Okay, I'm going to move on. My lovely host can continue answering questions in the chat. I'm going to jump back into some of those task verbs. So if you are asked to design an experiment, these seem scary, but they're a really easy way to get points. If you are asked to design an experiment, make sure you identify independent and dependent variables and say what they are, right? Say, oh, the dependent variable, I would measure oxygen. And the independent variable, I would measure, the amount of sunlight this thing is getting. Identify your constants and your controls, and then specifically state that you would do multiple trials, you'd use a large population, something to kind of increase the authenticity of your experiment so that you're not dealing with just, two trials or something like that. And I think Mrs. Shoemate will talk about this a little bit tomorrow. Same thing with when asked to make a graph, but I just thought I would touch on it because it is another way to easily get some points. They're really easy points if you make your graph, right? Label the X and Y axis, scale it with units. If you don't have units, you'll miss the point. So if, your X axis is a measurement of, height or something like that, or like depth of the soil that they're doing the study in, if it's in centimeters or inches or something like that, make sure it's there. And it is guaranteed on the long FRQ questions. So you will have to graph. There was a year or two where they didn't because it was virtual, and I think that was too difficult to do. But you will be asked to graph, and Mrs. Shoemate will talk about it tomorrow night. Another tip and trick is to tackle short, easy questions first. So look through the FRQ and just find the ones that you can answer. As long as you answer on the correct page. So in your AP exam booklet, it will have it'll have like on the top and on the bottom, space for question 2. And on the next one, space for question 3. So you have access to all 6 FRQs at the same time. You do not have to do the long ones first. You can flip through and be like, oh, these short ones, I know this, and tackle those, get those points, right? Because your big FRQs, yes, they're big, but you also have 4 short FRQs. So if you get your 4 short FRQs and you do well, then you can take a little more time on the long FRQs and don't feel like you're in the weeds. Someone mentioned this in the chat earlier, so I don't need to necessarily say it, but just make sure you use complete sentences. If you use bullets, or if you use like, if you write the word because, B/C, they will pretend like that word didn't exist. So do not use shorthand. Do not use real words, full sentences. Another big one that AP readers really, really appreciate is when you don't add fluff. So like in that last FRQ where it says, identify where this is happening, you don't have to say, I first remember learning about eukaryotic cells, and I know that eukaryotic cells have a nucleus, and that's where they hold the DNA. So, blah, 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 nucleus, right? Like that person's reading your prompt and they want to just find the answer. So, don't restate the question, don't start rattling off a bunch of things, and don't give an example that's not relevant. So, stick to the prompt. That's why we say ATP, answer the prompt. All right? Lovely. The other one that I love, and it's not a requirement, but I just think if an AP reader can read your stuff more easily, that you're just going to do better. It's just how I feel. So the paragraph on the left is the exact same set of words that are listed on the right. Which one's easier to read, left or right? Go ahead, tell me in the chat. Which one is easier to read, left or right? So look at the slide. What is easier to read? The one that I wrote on the left and the one that I wrote on the right. Love it. The left would make your day. Oh, yes. So again, it's not a requirement, but if the FRQ has A, B, C, D, yes, you can skip lines. If it's A, B, C, D, label it so that they know, boom, I'm grading A, I'm grading B. You can use one word for the identify questions. All right, really good. Thanks, everyone. And then I mentioned this earlier, don't contradict yourself because some students will be like, oh, well, if I say it one way, because when you're stuck between two options where you're like, oh, I'm pretty sure, translation happens at the rough ER, but also it could happen somewhere else. So I'm going to write it one way in A and one way in B and hopefully get the points either way, like get 50-50. But you just can't contradict yourself. They won't give you the points at all. So this is an example of contradiction where identify the relationship between a dog and a tick. So the relationship between a dog and a tick is parasitic because both benefit. And then they identify it in one word and say parasitism. And so it's like, no, you can't say that parasitism benefits them both. So you're contradicting yourselves. You wouldn't get the point. It would be kind of sad. Yes, we're talking about FRQs right now. Just some tips and tricks. One of the last things, I know some people can't help their handwriting, right? Like there's just some people with better handwriting. I don't have great handwriting. I'm left-handed. So if any of my students are here right now, like I'm left-handed. I try my best, but if I'm going really fast, it's really hard to read my handwriting. So I know it's a time crunch, but go slow because if your handwriting looks like this, it's so hard for them to read and they're going to try to read it. They'll pass it to like several readers and be like, can you read this? I can't. So just take your time and read and write as carefully as you can. Yes. All right. That was it for my FRQ things. We're going to take a quick break and do some practice multiple choice questions. So I'm going to go ahead and get up my some quizzes with that music that you all love. So I'm going to grab Unit 6 because that is something that I'm seeing a lot of people. All right, I'm going to put the quiz in the chat for you all. All right, quiz is up. We'll look at your questions as well. right. If you were with us yesterday, we did these first few. So we're going to go through the first couple pretty quickly. DNA replication is semi-conservative. Nice job. All right, uracil, very good. There's no thymine in RNA. There's uracil instead. Next. Yeah, we did do these yesterday. So I'm going to go through these first 5 super fast so we can get to the 18 total. Okay. So yeah, flow of information, DNA, RNA, protein. And again, we did helicase. That is what unzips. All right, this one was topoisomerase. I see a lot of you just not answering them because we already did it yesterday, so totally understand that. And here's the new one. Go ahead and answer this. Wonderful, 97%. You are all amazing. Those red in triplets called codons. Very, very good. Let's keep going. Okay, so this is a harder question for sure. DNA is synthesized in a 5 to 3 prime direction. So it looks like 24% of you said 3 to 5. So what I'd like to do in this moment, because that's like a decent split, I just want to draw something for you. Hold on. I'm going to stop sharing for just a moment. All right. So if you have DNA, right, your 5 prime end is going to be where your phosphate group is. I know it's showing it to you backwards, but hopefully you get the idea. Here's your 5 prime end. You're going to have your sugar here and then your base here. So your 3 prime end is right here. Okay. So 5, 3 prime. So the next new nucleotide is going to be added to the 3 prime end. So 5 prime to 3 prime. So a 5 prime, I'm going to draw a 5 prime phosphate. Again, I drew that really fast, but this is another really good test taking strategy, everybody. Draw it. Okay. So here's my 1 nucleotide and its 3 prime end. And the 5 prime end of this new nucleotide is going to be added to here. Okay. So the 5 prime end of this nucleotide is added to this 3 prime end. So that's why it's 5 to 3 prime end. Someone asked a really good question. Why are they called 5 prime and 3 prime? So when you if you took AP Chem, you might have learned about this, but in chemistry, we number carbons And so if we look at if you're looking at an organic molecule, your first carbon next to an oxygen is called carbon 1. And so basically the carbons of your sugar molecule are labeled 1, 2, 3, 4, 5. And so the phosphate's attached to the 5' end. It's okay if you didn't take chemistry. It's totally not a problem. But sometimes it's nice to know, like, why 5', why 3'? That makes no sense. But it's all about just this phosphate is attached to the fifth carbon over here. And then the third carbon over here is what the new 5' end will be attached to. So 5' to 3. 5' end to the 3' of the new one. So I encourage you to draw stuff out if you're feeling like, I just never can get this 5' to 3' thing. Draw it out. Draw out your nucleotides. Have it make it make sense for you. All right. So hopefully that helped. I know it was backwards. I apologize for that. Let's keep going. Oh man, let's go. 85%. Really good job. So the GTP cap on one end, the poly A tail, again, it's just a bunch of adenine bases that are added so that when that RNA transcript goes out into the cytoplasm, the poly A tail can get eaten up by enzymes and it's not going to affect our message. Really good job. All right, so this is asking about what picks up specific amino acids and carries them to the ribosome to recognize the appropriate codon. So your answer is going to be tRNA is picking up specific amino acids and carry them to the ribosome. Carrier proteins looks like it was something that popped up. I'm not sure what that's from. But I also want to just let you know, I saw someone in the chat just a second ago that asked if these are going to be similar to what the AP exam will show you, and they're not. We couldn't write AP style questions in this program because if you've taken an AP practice test or anything on, oops, what happened to my screen? Why am I so big? Someone help me. No. Can my teacher host help me? I did something where my screen is too big. Nope, that's not what I wanted. Shoot. Return to previous state. Anyway, okay, thank you. Sorry, I was distracted by the technology not working. So if you've taken an AP practice test or anything on AP Classroom before, you've seen the multiple choice questions, hopefully. They are longer, right? They can be set questions where it's 3 or 4 questions based on one big scenario, but they can also just be singlet, and sometimes they'll be easy, but a lot of the times they're not going to be a one-word answer option, okay? It is a college level class. It's very rare for them to be a one answer option. Yeah. And then someone asked about why we can't use the real practice test questions. So some of the practice, there's only one publicly released practice test, and it was from 2013. And the exam has changed pretty drastically since 2013. So we are using FRQs because those are free and online for anyone to look at. But yeah, we're not allowed to show any other, AP style questions. Someone did the 2013. Yeah, the 2013 was still really old, where they were still testing on body systems and plants pretty heavily. transcription and translation, they happen in the cytoplasm in prokaryotic cells. So read carefully. I see some people just said in the nucleus. That would be for a eukaryotic cell. Remember, you and I are eukaryotic organisms, and we have nuclei. Okay, so eukaryotic nuclei. Prokaryotes do not. All right, so operons are tough. I know that. So it's asking about what type of operon. It's an inducible operon. Operon is often off and repressed because the repressor is always active. This was that example we showed last night with the lac operon. I want to tell you, operons are hard, right? Inducible or repressible. They're tricky, totally. Again, that's one of those things that I remember as a student. I would draw out, right? I would draw out both operons and say, okay, well, if this one has a repressor, right? The repressor's on there. And if the repressor is always there, it's repressing. It's not letting gene expression happen. And so it's hard stuff, but just, diagram it out for yourself. And then also, I know I said this is unit 6. I said this yesterday that like, if there's a particular topic that just is so hard for whatever reason, there's something strange where you're like, operons, I can never wrap my head around operons. That's okay. Like we're at the point now where you can give yourself grace for that and you can move on and work on something else that will make sense by Wednesday, right? Like chi-square or something like that. So if there is something there, you can be like, oh, wait, I can give operons another chance, right? Okay. So don't be discouraged, okay? We got this. Right, great participation, everyone. Yes, the answer is B, transcription factors. So transcription factors are generally proteins that kind of help bend a eukaryotic gene around so that you can have your promoter interact with RNA polymerase. It has the word transcription in it. And so sometimes that can help remind you that that's there to help regulate gene expression, that like it's a factor that needs to be there for transcription to occur. And so sometimes in a way to, be more efficient in a eukaryotic cell, the same transcription factors can regulate different genes. And so they're kind of relying on cell signaling and things like that to actually go regulate that gene. But yeah, I totally understand that is a hard question as well. I don't love this question, just FYI. I didn't write all of these, but this is one of those like gotcha questions where if you don't know the one specific tiny fact, so it's not testing your application skills, it's not testing that college level understanding. Let's just reveal the answer. It's going to be your promoter. Oh, you're amazing. 81% did the promoter. All right. See, we got this. Okay. Okay, go ahead and answer the next question. right, in the interest of time, I'm going to keep speeding these up. So 14, when does insertion mutation lead to a change in the amino acid sequence? So I see why a lot of you said C, that just sometimes because some codons code for the same amino acid, but with an insertion, that means one nucleotide came in, so it was inserted, and you already have all these codons that are very specifically being read to make specific amino acids. But it's like if you're waiting in a long line to get lunch at the cafeteria, and like one person cuts in front of you, that frame shifts everybody else behind you. So insertion really is pretty bad because the whole thing shifts the reading frame. I agree with you that yes, some codons code for the same amino acid, and that's why things like substitution isn't a big deal. So like if a T is substituted for an A, it typically isn't like the biggest of deals because there might be a chance that it's still the same amino acid. Yeah, I mean, it's really unlikely if a frame shift happened and somehow every single amino acid after that was unchanged, but it would be, yeah. So that's why insertion is just a little bit major. We get a whole frame shift going, but really good job. Really great participation, everybody. All right, there's an error in DNA replication, is when mutations can happen. Generally, if there is an mRNA issue, that's kind of past the level of like a mutation. But it is, I guess it could be possible. So I see why you said B, but I don't think that you would be asked a question in that specific way. It would be more about what's happening in DNA. All right, we'll do a few more questions. All right, transformation. I was worried people would say A. Okay, so if a bacterium picks up a floating piece of DNA in its environment, it's not conjugation, because for conjugation, there needs to be another bacterium right next to it where the conjugation comes. Like you've probably seen the picture of like the two bacteria, and then it looks like a bridge across from them. That's conjugation. So the only reason that's wrong is that this is talking about a floating piece of DNA in its environment. When we think of bacterial transformation, you're like, wait, that was way harder than just, bacteria floating around. But in nature, that can happen, right? That can happen, that a plasmid enters into bacteria. So, all right, let's do one more. Let's do this question. Ooh, 50-50. All right. Oh, that's weird. It has the same answer twice. that's an error in the question. That's a good thing to know. So in gel electrophoresis, it's going toward the positive end because DNA is a negative molecule because of the phosphates on the backbone. And so if you're migrating further along in the gel, you're going to be shorter. So B and D are both right. I apologize for that. That's kind of silly, but there were two. And again, if any of you during these quiz questions are feeling, like I didn't know any of those, that's okay too, because on the actual test day, you are given so much information in the problems, right? Like you really don't need, you don't need to have a ton of things memorized. So I'm going to give everybody a 3-minute break. Okay. So go to the bathroom. I will set up a 3-minute timer real quick. I'm going to put that in the presentation. So go ahead, 3 minutes. We'll see you back here. That's the end of our timer. Okay, wait. Hold on. Okay, wonderful. Okay. Hi everyone, welcome back. My screen keeps getting messed up. Can one of my student hosts help me? One of my student hosts is going to come on here and do a quick icebreaker, think about it question just for fun, just to kind of get our brains at ease. So come on over.
Hey guys, I hope you had a good break. So the icebreaker for today is, what's your favorite mythical animal? I'll start first. I guess my favorite is a phoenix.
So you can respond in the chat. Let us know. Just kind of a fun, funny question. Get our minds feeling happy, ready for the next part. Oh, unicorns. Do our other student hosts want to share? You don't have to show your face. You could just unmic. Do you have a favorite mythical creature?
I sent mine into the chat, but I am a hardcore fan of the unicorn. I don't know
Nice.
if anyone else read the Magic Treehouse story where they end up like finding a unicorn, but that one was like one of my favorite stories ever as a kid.
Oh my gosh, I love that.
For me, I think like this is a pretty funny one, but I think gnomes are probably my favorite. I think the most important reason is like besides being like little stupid like decorations you see, they're actually known to symbolize good luck. So yeah, I feel like that's really cool to just cherish and know.
I love that. Thanks, team. Thanks for sharing. Magic Treehouse, yes, I do too. I love that. Okay, let's get into the science practices, okay? I know the first hour was kind of like tips and tricks, FRQ. We did Unit 6 multiple choice questions because I saw in the Q&A that people were asking about a little bit more coverage on Unit 6. And then let's get going on our science practices. So as far as, what are the science practices and how well do you need to know them? It's not that you need to know them by name, right? They're not going to say, how well do you know science concept, practice number 1. However, we as teachers know what each of these science practices are, and we also have been told what the exam weighting will be. So take a look at this and just tell me in the chat 1 through 6, which practice, actually 1 through 3, because that's what we're covering tonight. Which of the 3 should we probably spend the most time on tonight based on its weighting? Good. Love it. Yeah, so you're already seeing the exam weighting is pretty high for concept explanation, which just means, can you explain concepts? Like they're going to present a concept to you and you're going to have to explain it. Or the answer options in multiple choice will be explanations and you will have to pick the right one. We'll talk about strategies on that in a minute. Mrs. Shoemate's going to cover argumentation tomorrow night. So you'll get a lot of argumentation practice with her. So what is concept explanation and how does it look on a multiple choice or free response? So in a multiple choice question, what they would do is kind of give you a model presented in a written format. So they might give you a little story about, there's this organism that can take in oxygen and release carbon dioxide and blah, 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 blah. And you'll have to choose which description or explanation best fits the process that was described. So it's a lot of reading in a multiple choice because you have to wrap your head around what they're saying. But then you really are just choosing what's the best explanation for this. Free response, this science practice is in 1, 2, 3, 4, and 5 for science practice 1. So almost all of the free response questions have you explaining or describing something. Science practice 2 is visual representation. So you'll see an image or a diagram, and you're going to have to describe that concept within what the diagram is saying. And then free response, you'll see this in question 5 and in question 5. So that's also just something nice to know that like, if you're really good at the visual stuff, flip over to question 5 and get that done, right? Like do that one first and get your 4 points. Science practice 3 is questions and methods. So this is anything related to scientific investigations. And they might ask, what's the best control for this experiment? They may lay it out in a multiple choice, or they might say, what would be a null hypothesis here? And I'll talk about null hypothesis in a minute. And the free response 1 and 3 will focus on this practice. And then half of the points for each question will also be assessing this. So like it's a decent amount for questions and methods as well. I should have shown this disclaimer earlier, and we kind of mentioned it with some questions in the chat, that we are only using questions that are publicly released. We are not allowed to, and nor it's not, within, it's against our integrity to do otherwise, right? So we are only using publicly released questions, and that's what makes these tests so hard, right? We don't have access to like what the people wrote. So we can just do our best and try our best. All right, so let's dig into science practice number 1. You're going to see the describe and explain task verbs a lot. They will give you, you'll have to be looking at a biological concept. So like I said, something like photosynthesis, right? Can you describe this one component in photosynthesis based on the information they're giving you in the prompt? It's going to be very rare that you'll see a question that just asks you for information out of the blue. Generally, every question comes with some sort of context. They'll give you a little story, a little investigation that it's about. And so you're really very rarely answering a question just on your own. Okay, so this is a free response. Rashi, you want to take this one over?
Yep.
Thank you.
So I'll read out the question, and then you guys will have 6 minutes to go over or to practice it on your own, and then we can go over it as a group. And this is from the 2022 practice exam, or actual exam. Existing isolated brook trout populations in Newfoundland, Canada were once part of a larger population that was fragmented at the end of the most recent glaciation period about 10,000 to 12,000 years ago. Researchers invested 14 naturally separated stream populations of brook trout. They found that populations are all genetically distinct and show differences in morphology. So there are 4 parts of this question, and you guys have 6 minutes to answer it, and then we'll be going over it as a group. Good luck. Time's up. So let's go over the answers to these questions. And before we do that, I'm also quickly going to go over what the questions themselves were asking. So the first one was, or part A was, describe the prezygotic barrier that results in these genetically distinct populations. So here the key term is describe. You want to not just mention what the zygotic barrier is, but also just like relate it back to why it is causing these genetically distinct populations. In the second, or part B of the question, it's asking you to explain the difference in fitness between longer finned individuals and shorter finned individuals. So if you just said there is a difference in fitness, you are not going to get the point because you're not explaining how it relates back to the prompt. Part C asks you to predict the likely impact on both populations. And so this could just be like 2-3 simple sentences. But the main point here is that it's asking for the impact on both populations. So you want to make sure you're not just covering the longer finned individuals, but also the shorter finned individuals. And then part D is asking you to justify the researcher's claim. And so here's where you'd provide evidence and prior background knowledge to describe your answer. And then now on the next slide, we'll start breaking down the specific answers. So for part A, describing the prezygotic barrier that results in the genetically distinct populations, the answer is that there's geographic isolation, and so there's no longer a gene flow between the populations. They become isolated, they're only reproducing within their individual groups, and so they become genetically distinct because there's no longer this intermingling between the populations. For part B, explaining why there is a difference in fitness between longer finned individuals and shorter finned individuals. Well, it tells you in the prompt itself that brook trout with longer fins are able to swim faster than brook trout with shorter fins. So if the prey are also becoming faster, the individuals with the longer fins are more likely to capture these prey and reproduce because they have evolved to become faster, and they'll be able to keep up with the now faster prey as well. For part C, there were 2 options you could have given for your prediction. The first is that the 2 populations will become separate species because they've remained isolated for so long, or that they'll continue diverging either behaviorally, morphologically, or genetically. And this is because these populations, as it said in part A, have become genetically isolated, no longer intermingling with the 2 groups. And so if they continue to be isolated, it'll just be the same traits reproducing, and they'll split into maybe perhaps 2 separate species. And then finally, for part D, justifying the claim that there are more genetic differences between any 2 current brook trout populations than there are between any single population currently and the ancestral brook trout population from which all trout are descended. So there are 2 possible answer choices. The first one is each single population has accumulated mutations or experienced genetic drift, and this is distinguishing it from the ancestral population. But the mutations each population accumulated are likely to differ because of different selective pressures. The trout brook populations are all living in different areas. They have different selective pressures. Therefore, they have different mutations. And so each population becomes very different from each other. But they're still linked to that ancestral trout brook population closely. And so they are genetically closer to that ancestral population than the other populations who are experiencing different selective pressures. Another possible answer choice is that allele production as a result of random mutation and genetic drift or selection by local environmental conditions has resulted in a collection of alleles unique to each population. I'm seeing a lot of questions on asking what part D is asking. So I'm going to go over what the prompt is once again. So here it's saying like there is a claim that between any 2 current brook trout populations, so maybe on this level, there are more differences here than between any single current population and the ancestral brook trout population from which all trout are descended from. So there are more differences at this level than there are from here to like here. And it's asking you to provide reasoning to justify this claim. And so the two answer choices here that are discussed, the first one is that each single population at this level has multiple mutations in genetic drift, but all of these mutations are different from one another because of different selective pressures. These populations are in different environments, and so they have different pressures. And so they have so many different mutations at this level compared to each other versus this level and this top level from their ancestral population. The other answer choice for part D was talking about allele production because of random mutation, that each one of these groups has a different set of mutations. So this group has mutation 1 and this group has mutation 2. There's only one mutation difference between here and here, between like the current population and the ancestral population. There are 2 mutation differences between population 1 and population 2. And so that is how you would answer this FRQ.
Thank you so much, Rashi. That was so good. And I know that these FRQs can seem confusing and scary, but you did such a good job explaining the scoring guidelines and just really helping everybody through it. So I love it. I see some questions as well from people, and I agree that when you first look at something like D, it's super scary and you're like, what are they even asking? So I'll give you a AP tip where if there's something, a prompt or a part of a question that you just can't quite wrap your head around it, draw a picture. Okay. I am not amazing at drawing pictures. So I'm going to move myself over. So D is saying, this is the population that the prompt is talking about. They're saying that there are more genetic differences between the 2 current brook trout populations. I drew them differently. So like these ones are colored in purple and these ones are not. So these are both trout and they're living in a similar area, but the researchers are saying genetically, these are more different from each other. So this shaded in trout population and the non-shaded in trout populations are more different genetically than all trout compared to the ancestors. I know I wrote backwards so you could all see it. It's badass. I try my best. So they're saying, okay, the current population between these 2 things are more different genetically than all trout that ever were compared to their ancestor long ago, where you would expect that like genetic differences between an ancestor and all current trout should be pretty big, right? Like things have changed over time, but they're saying, no, this is something really interesting. These are, oh, it does show up for you. Anyway, these are more different genetically, living in the same stinking pond or wherever they're living or river. They're more genetically different here than the ancestors to all current trout. Oh, it's flipped. Oh my gosh. If I write it backwards, it shows. Thank you so much. Well, I don't have to write backwards now. That is such good news, everybody. Oh man, it's 8:30 here. I'm trying my best. Next time I draw you a picture, I will just draw it normally. I was like so proud of myself for writing backwards. All right. So someone's asking, but why are they more different? If you go back to the prompt itself, I know sometimes we get into the weeds of looking at the prompt itself or looking at the thing, but the prompt tells you that there's they give you the story, right? They say they're looking at 14 naturally separated stream populations. Oh, that's why. So they're originally a part of this place in Canada, and they're looking at 14 naturally separated stream populations. So they're living in a stream, but they're naturally separated. And that's why they're saying they're more genetically different. And so that kind of was explained in A through C too, that when they're different and that far apart is where genetic differences can happen. All righty, let's keep moving. You're doing so good. So I'm going to skip through this and get to the next one. Okay, let's just take a look at a picture and think about, what is this? What biotopic is this? We're in concept explanations. If you were given an image like this, what word or topic would come up in your head? Love it. I'm seeing osmosis. You're absolutely right. So osmosis, where we're talking about the movement or diffusion of water across cell membranes, mainly through these transporter proteins called aquaporins. They're facilitating the diffusion so that water isn't moving through the membrane fully on its own. So this is all about osmosis. We're talking about passive diffusion. I love it. So sometimes it's really helpful if they give you a diagram before you even look at the question and be like, what topic is this? I know this, right? And just kind of get your brain to the right spot to be able to answer the question. Okay. So that's what we did there. We looked at an image first and we started pouring out what we knew about those things. So, thinking about realistically and in our lives, why can't humans be given IV fluids of pure water? Good. Yep. Ooh, I'm seeing some differing answers here. I have some people saying our cells would lyse. Some are saying they would explode. Yeah. So again, draw yourself a picture. Osmosis was my favorite time to draw pictures. And so if our cells were in pure water, our cells aren't pure water, right? We've got all sorts of stuff in our cells. We have tons of solutes in our cells. We've got sugars, we've got potassium, we've got all sorts of things, right? And so if this is our body cell, then being injected with pure or being surrounded in pure water, that would not be good, right? Water is going to move into the cell and cause it to rupture. And so I saw a lot of people saying that, but again, if things are confusing to you, draw yourself a little image. And I know you're thinking like, "Oh, I wish I had time on the exam," but you're going to waste less time in the spins. Like if you've ever been in a test and you're just like your mind feels like you've spun and spun and spun and you're lost, this will help you focus. This will help you zoom in on what you do know. And at the end, if you're like, I'm spending too much time on this question, I'm guessing C, then that's okay too, right? You have 60 questions to get through. So keep going and find the questions that you know for sure, right? All right. Let's keep moving along and we'll take a break soon. I know we're an hour and a half in. Okay, so here is an osmosis question. It says the figure shows two containers interconnected by a tube. The tube is partitioned by a membrane permeable to water, but not sucrose. So it's permeable to water, but not sucrose. What does that mean? So go ahead and respond in the chat. Oopsie, I just gave it away. So again, it's partitioned, which means right here, there's a little thing right here that it's permeable to water, but not sucrose. Permeable to water, but not sucrose. What does that mean? Good. So sucrose cannot go from X to Z, but water can. So that's what it's saying. So if it's permeable to water, that means water can go between the two sides, but sucrose cannot. And so if your teacher did that lab where you did the dialysis tubing, it's like that clear tube, and you wanted to see if like was starch leaving, was sucrose leaving, was water coming in, and you did all that kind of thing. You might have done that lab like hands-on, but there's really good, really easy virtual dialysis labs, and that's what I had my students do because it's a pretty quick, easy lab that you all can understand. All right, so I've got a question for you here, and I know it's a lot to read, so I'll read it to you. It says, beaker X contains a 15% sucrose solution. Beaker Z contains 11. And we see that in the image here. Assuming that temperatures and pressures are equal on both sides, which one of the following statements best describes what will happen in the system? And it's best describes. A, water will move from X to Z. B, water will move from Z to X. C, water will move equally in both directions so that there will be no change in the system. D, water will move in both directions, but the net flow will be from X to Z. Or E, water will move in both directions, but it will be from Z to X. So I should have made a poll for this. Sorry, I forgot. So A, B, C, D, E, you can say it in the chat or just think it in your head. It's totally fine. Okay, so hopefully you said E, that water will move in both directions. That's actually a really big misconception with osmosis, is that like water only moves when it needs to, but water's always moving in and out. It's just kind of like if you have your windows open and air, like the wind comes in and out all the time. There will be times where there's going to be bigger gusts of wind, but it's not a closed system all the time. So there is a net flow. It will be from Z to X. If you want to start throwing in vocabulary, I already saw some people in the chat saying like, well, one is hypertonic, right? So water is going to move toward the area that has the highest concentration of solutes, which is that 15% sucrose. So if you need to draw in dots to help you with that, like I would tell my students, okay, find the thing that has the most solutes and draw a bunch of dots to represent those solutes so you can visualize that. And water is going to go toward those dots. It's going to try to dilute that situation. Okay, water potential and tonicity. I am not going to do the math tonight because Mrs. Shoemate has that planned for you tomorrow, but I wanted just to kind of show you how, yes, you need to know osmosis, but then also there's this whole other layer of doing water potential mathematically. All right, but I do want to talk about the concept of water potential. So what is water potential? Go ahead and read that. All right, so water potential, if you ever took physics, and it's okay if you didn't, but you might have learned about potential energy, right? So like if I held a book really high up, right, it has potential to do work, right, in fall. So water potential has to do with that this possibility to be able to do work and move. And so pure water has no solutes and has a water potential of someone hacking my computer and putting that song on. Anyway, so water is 0 for water potential. But as solutes are added, the water potential decreases and becomes negative. Okay, so we go from having it's called a psi. This is the symbol for water potential. It kind of looks like a trident, right? So that's the symbol. I think it's from like Poseidon, right? Like the water that would have been a good mythical creature actually. Anyway, so as solutes are added, water potential decreases. It becomes more negative. So let's do a poll. I'm going to type one in real quick. Think about your answer option. Poll is live. I like what I'm seeing. Very good job, everybody. All right. It is the right. So this has more solutes. So it has a lower water potential. So it would be a negative number, more negative, right? Like let's call this -5 on the right. And this one's -2 or something like that, right? Because it has less solutes. It's going to be closer to pure water. Excellent. This is another way to think about it. If water potential is difficult for you, I like that. I like made this number line because it helped made when I was a student. This is how I thought of it. Pure water is 0, right? We don't get into the positive numbers until we get like pressure potential. So if they give you like a closed system, which I've never seen on a test, they might give you like, hey, the plant has a pressure potential of 4 or something like that. But I've never seen anyone ask you to calculate pressure potential. So anyway, if it's an open container, like a cell in an open container or something like that, and more solutes brings you negative. Okay. And then water, it's kind of like a water slide. Water will move from that 0 potential to something that's more negative or more hypertonic. If you were to throw that hypertonic word on there, right? So think of it like a water slide, and you've got this number line from 0 to negative numbers, and water is going to flow from those zeros or for like less negative numbers to something more negative. So I just thought that was an easy way to remember it. Hopefully that helps you. All right, let's do a practice problem. I'm going to make another poll real quick. This is not something you have to calculate, okay? This is just think about those numbers and how they're different from each other and which way water would move. So go ahead and answer the poll number 4. So a cell with a water potential, here's my cell, -2.5. I'd write it on the exam. I'd write it right there, -2.5. It's in a solution that's -0.7. Which way will water move? Remember, it's going to move towards something that's more negative, and so it's going to go into the cell. It's moving from high water potential of 0, which is kind of weird to think that 0 is high, but it is, high water potential to low water potential. So if you think of it this way, -0.7 is closer to 0 than -2.5. That could be a way to think of it. All right. I'm going to skip the math side step because I looked at Mrs. Shoemade's presentation and hers is fantastic, and we don't need to go over this because she's got you covered. Okay. And I'm going to skip this too for time purposes. Okay. Same thing here. All right. Do we need a break? I think we need a break. I think everyone needs to go to the bathroom real quick. Yes? Good. Okay. I am going to put timer on for just a few minutes so that we all know when to come back, okay? Go ahead, take a break. Go to the bathroom, do your thing, okay? I'm going to move my face out of the way, and we're going to take a 3 to 5 minute break. I will see you back at 8:50. Okay, everybody, we are back from our break. Let me get moved over here. I hope you had a good couple minutes. I actually ran upstairs and was able to tuck my kids in and give them kisses and say goodnight. So I hope that you all feel ready to finish this up. Let's go. We have an hour. We can do this. Let's see this. Okay. Another topic that I wanted to talk about, we didn't have much time to talk about it the other night, is this idea of epigenetics. Now, epi means, if you've ever taken anatomy, epi means like the surface level. So like your epidermis is your skin. So think of it as like the surface level of the genome, right? It's not changing the genome necessarily, but it does affect gene expression. So epigenetics is things that happen on the surface level of the genome that can affect gene expression. So this diagram shows a gene in red right here. And it's wrapped, these DNA is wrapped around these histone proteins, which you don't need to know about histones, but basically when these methyl tags are put here, you can see it's got a methyl group right there. And when they're put in here, these methyl tags kind of hides the gene and makes it inactive. It doesn't mean it's erased, the DNA is still there, but it's inactive. So I want you to just think about for a second, because again, we're doing science practices. Why would methyl groups, knowing that methyl looks like this, it's a carbon with 3 hydrogens, why would methyl cause DNA to become less accessible? Why does it make DNA come into this like ball and be inaccessible? So think about that. You don't need to know histone tails. Don't worry about that. Just think about methyl, that shape of CH3, and why would it behave in a way that hides things? Yay, Nadia, you got it. All right, so yes, so methyl is nonpolar. So it is a carbon with 3 hydrogens. And this kind of ties in the stuff you probably learned with protein folding, that if you have an amino acid with a methyl group, it's nonpolar. It's going to behave in a way kind of similar to fats, like they want to hide from water. And because DNA is in this watery environment, it's going to hide. It's a really big jump for you to be able to get to that explanation, but it's one of those things where it kind of highlights, something you learned in the beginning of the year, but now it's bringing it up in genetics. So when you see carbon and hydrogens and you don't see any oxygens, or you don't see any phosphates or anything else like toward the right-hand side of the periodic table, it's going to most likely behave in a way that's nonpolar. And so it's like going to hide. Okay. Again, everything we do here, just remember it's one topic out of all the topics. And so if there's a couple topics that you're just really struggling in, give yourself grace. It's going to be okay. The fact that you're here and you're showing up. I do just want to remind you too that like you're in charge of your brain and you're in charge of your actions. So if you're spending more time having side conversations and not really testing yourself on this stuff, then that's only going to hurt you later. So I appreciate those of you who are really trying and helping each other in the chat, but just remember that, keep conversations about biology or about fun stuff, and we'll all be good to go. Okay. The next concept I want to talk to you about is evolution and phylogeny. So this is unit 7. And what I've got here is a chart showing amino acid positions. So it's not the whole strand of amino acids. It's, out of 100, we're looking at position 39 through 53. And we're comparing animals. So horse, donkey, rabbit, snake, turtle, whale. And if you look down vertically, all of them have the same amino acid for position 39. They all have B for 40. They all have C for 41, so on and so forth. So like the first few amino acids, they're all very similar. But then you start to see, oh, look, at 44, horse and donkey have F, rabbit and snake have Y, turtles V, whale is Y. So you start to see there's some differences, and we can start to see, hey, who's more related based on this? Where could we put them on a tree based on this? And so I want you to think about why we can use amino acids to compare evolutionary relationships. So why can amino acids be used? So amino acids can be used because amino acid sequences come from DNA sequences, right? And so ultimately, we're comparing DNA sequences in a way. Yes, so it's a very specific thing. They link back to DNA. I love a lot of your answers. Really, really good. Awesome. All right, let's take it a step further then. Poll number 5. Which organism, I got to write it. Hold on. Poll number 5. Which organism is most related to the horse? So you're using this based on the data. Always be careful that you're using things based on data, not based on your previous understanding. So which organism is most related to the horse? So the poll is open, but you can also always use the chat. Yeah, wonderful job. As you can see, over 90% of you are, oopsie, the donkey, right? So we could look through and say, hey, look, they only have one amino acid difference, number 47. So here's an AP question for you. You don't have to say it in the chat, but I want you to think about it. If you had to defend your answer or use evidence, you'd want to specifically say, based on the diagram, the horse and the donkey have the exact amino acids at positions 39 through whatever. Or you could say they differ in only one amino acid out of this group of 13 or 14, right? So you'd want to link it back to the data and say, based on this diagram, this is what I'm seeing, and this is why I said that. Okay, this is from the 2019 exam where they gave, and I showed this to you last night, but basically it's showing the divergence of mitochondrial DNA sequences. So it's saying like, how different are they? And that's another thing where sometimes they'll use a word like that and you're like, gosh, divergence. I mean, it sounds like things are separating or things are different. Like it might throw you for a loop, but just, you can do this. Like try to use common sense if you can. So last night I talked about the importance of finding the out group. And if you look at, for example, the gorilla, right? If you compared the gorilla to everybody, you could be like, all right, well, my gorilla and human are only 10. My gorilla and orangutan are 16. Then we've got 18 and 10. So it's like, okay, 10, 16, 18, I get it. But you want to just look for the biggest numbers, right? And so I see 18, 18, 18, and I see another 18, right? So it looks to me like the gibbon is the outgroup. And in fact, on this prompt, they did give the gibbon as the outgroup for you, which is a possibility, which is so cool, right? You don't have to always, just draw something from scratch. And in the last few years, they've also provided the actual tree so that students aren't having to build a tree. And I think they probably are doing that because it's easier to grade, to be honest. So what I want you to think about, if you want to draw out on your piece of paper how this tree is drawn for you. So it's one line going to the right and then 4 extending lines with the gibbon at the bottom. So like draw that out for yourself and put gibbon at the bottom. And then I'm going to go back to the data and have you try to place the animals. Okay. We're not going to do the whole FRQ. We're just going to do the tree. Okay, hopefully that was enough time to let you draw your tree. So go ahead and take a minute or two. I'm going to hide myself so this can get bigger. All right. Go ahead and try to build the tree. Sketch it out. right, so I'm going to walk you through this a little bit. We talked about the gibbon being the out group. Your next thing is going to be to try to determine who's the next most different, right? Who's the next organism that seems to have high numbers? And my eyes go to, I see 17, and that's between the orangutan and the chimp. And then I see the 16 again, and 16 for orangutan. And so in my mind, I'm like, okay, I think maybe the orangutan is that next one. And I'd put it on the line. And then your last two that are even an option. Oh, wait, no, just kidding. We've got three, right? Human and gorilla. Yeah. Then your next one, try to figure out, okay, well, these are all being compared to a human. So we only have chimp and gorilla to think about. And we look to see who is the next least different. So I'm just going to click ahead for time. From top to bottom, we'd have your human chimpanzee. Those would be interchangeable because at this node right here, it doesn't matter what's on top or bottom. But because all the data was compared to a human, you can put your human on the tree. So human and chimpanzee on the top, and then you've got your gorilla and then the orangutan like I talked about. Okay. Again, if you think about this, the construction is 1 point, the reasoning is 1 point out of 4. So if this is one of those things where you're like, these trees, I always mess it up, then it's 2 points, right? Out of all the points. And you'll get those points back somewhere else. All right. So no need to be too stressed. Okay, so we've got visual representations. It's science practice 2. I know we only have an hour, but like I said, that first one, the concepts and explanations, is like 30% of the exam is that kind of stuff. So that's why we spent so much time on it. So for science practice 2, it's about visual representation. If you look at how they describe it, it's going to be giving you something visually. There's going to be some sort of a model. Sometimes it might be mathematical, but it could be a diagram as well. So I'm going to stay off and hidden, and we're going to have Samantha come and do this FRQ with all of you.
All right, so this is an FRQ from last year's AP exam, and take a couple of seconds to just look at the graph. And you might want to take a picture of it to help answer the questions on the next slide. Okay, so here are the questions that go with the FRQ. And just take a few seconds to read through it. And let's take 5 minutes to answer this.
Oops, sorry. Sorry.
And that's time. Let's go over part A. So I'm sure most of y'all know that what makes a species invasive in a community is what makes a species invasive for a community is their lack of competition or natural predation. So if that same species is in a different community and its population is being regulated, that means there's some competition or other external factors. And if you got that point, good job, guys. So for part B, you mainly have to refer to the graph or the figure to understand this. And what they're asking is how the species PP1 has a greater effect on community B than community A if it's removed.
Oops. Oh no. I'm sorry. I refreshed my screen to fix something and it didn't work. Sorry, everybody.
No worries.
I was trying to give you the image. There we go. This is what I was trying to do. So that for part B, everybody could see the image. Okay, there it is.
Thank you. Thank you for that.
Mm-hmm.
So as you can see on community B, there's only two primary producers. And if you remove PP1 from community B, then PC2 won't have any energy to get access to. So it will basically have a higher impact on that community than community A. And that's it for part B. Yeah, and this is part C. Y'all, I know this looks long, but it's very simple. You basically draw an arrow to show where species I and B goes to. And it's literally in the first sentence of the question, species I and V that eats individuals of species SC2. So I think you know where to draw the arrow from there. And the last one, it usually wants you to have a bit more background knowledge. And for this, they're asking how human activities affect the toxins of the soil, right? So they show that the human activities could eliminate primary producers because pollutants, they are passed down from one trophic level to the next. Therefore, they can't be easily broken down. This causes most of the animals to have, or other organisms to have diseases or any mutations, thus reducing species diversity. And that's it for this FRQ. If you guys got the point, good job, guys.
Wonderful. Thank you so much. I see some talk in the chat about some of you are noticing that as you go from A, B, C, D, it gets harder. And that's absolutely right. So usually the first two are like a describe, identify, explain, but then C and D get into the provide reasoning or justify or predict. Okay. So let's see here. Somebody wanted to know, can you just say it reduces producers, which will affect the food web? Let me go back to the prompt real quick. you would want to explain, you could say it would reduce producers, but you couldn't just say and it would affect the food web. You'd have to go a little further right there, and be a little bit more specific by what you mean affect the food web. Okay. I also noticed in the chat and in the Q&A that after doing our first phylogenetic tree, some of you were like, wait, what? And so maybe I went too quickly. Here's another one. This is from, I showed this last night. I believe this is from 2015. And so this is the number of amino acid differences in cytochrome C, which is just a protein, among 5 species. So some of you were wondering about, why are some of the boxes shaded in, or maybe there's a dash. When you're comparing 2 organisms at a time, if you set them up in a box like this, you could double up, and I guess you could fill in all these things. But for example, like this organism E. ferris and this organism D. polyepis, we should only have to compare them once, right? So like that's why this spot is blank, because we already compared those 2 right here, and they have 21 differences. So you see that D. polyepis here is compared to E. ferris there. So we don't need to show it here as well. So ignore the blank boxes. Okay. I know it's a little unsightly and you're like, what's happening? But it's just that information is not necessary because it's already there. So you want to look at these tables horizontally, so left and right, and then vertically as well, because sometimes students look and they're like, oh, look, I see a 20 for E. africanus. That must mean E. africanus is the most different. But if I go down this line, I realize, well, actually, E. africanus is only 10 and 12 differences between the other 2, right? Between this G. gallus and A. forsteri. So it's maybe not E. africanus. It's maybe who E. africanus is being compared to here, which is D. polyepis. And then you can confirm it with, okay, well, look there's 18 differences with D. polyepis and gallus. There's 17 difference between D. polyepis and forsteri. And so that's starting to look to me, oh, and look, and 21 between D. polyepis and E. ferris. So right away, oopsie, hold on. I meant to put in the phylogenetic tree there. So just give it a second. It should show up in just a moment, I hope. Let's hope. No, come on. Sorry, everyone. I'm going to put that tree in there for you because we're going to build this tree and we're going to walk away confident in drawing a phylogenetic tree. There we go. Now you should see it. Okay. So now that we've got our out group, which is D. polyepis, I want you to go ahead and sketch this tree for me. And you're going to put D. polyepis here. This is the out group, okay? It's the one least similar to everyone else. It's the one that's, most ancestral, we'd say. Like here's the common ancestor at this node at the bottom. So this is the most ancestral. It has the most differences. And then you notice off to the right that we've got like a pair here and a pair here. And I've got good news. This node means that both of those pairs could be either or. So really now you just need to look at which 2 are most similar and what are the other 2 that are most similar. So take a moment and try to figure out which 2 are really similar together and what are the other pair of most similar organisms. So take a moment, find those pairs for me. All right, so I know that some of you were here on Friday, and I believe Ms. Shoemate might have covered this one as well. So it's okay if we have a little repetition and a little bit of redundancy because every night is different, right? So some of you might have attended Friday and some of you might not. So we look at who is the next most similar, and I like to go to the most obvious ones first. So I look at look at this 3. There's only 3 differences between A. forsteri and G. gallus. Like those are my pairs now. And I can put them here and here. So G. gallus, A. forsteri, doesn't matter which one, right? Here or here, but they are in pairs together now because they only have 3 differences. And then the other one I noticed is E. africanus and E. ferus, man, only 1 difference. So they are also paired up. So E. ferus and E. africanus are paired up as well. So what once is hopefully something that was confusing is now a little more clear for those of you who hadn't seen this yet. And of course, there's not an endless amount of FRQs that correctly align to the current exam because it changed several years ago. So you probably have seen these before, and that's okay too. All right, we're looking at a free practice exam from the 2013 release practice exam. This is a multiple choice question just that I wanted to throw in there for you. So take a moment and I'm going to make a poll for you to answer so we can clear up the chat a little bit. So I'm going to make a poll for this. For those of you who maybe can't read it if it's on your phones, it says, undersea landslides can disrupt marine habitats by burying organisms that live on the ocean floor. The graph above shows the size of a population of a certain organism that lives on the ocean floor. The population was affected by a recent landslide at the time indicated on the graph. Which of the following best predicts how the population will be affected by the landslide? A, the surviving organisms will evolve into a new species. B, the reduced population will likely have allele frequencies that are different from initial population. C, the population will adapt to deeper waters to avoid future landslides. Or D, the reduced population will have a greater number of different genes than the initial population. We are at a 100% accuracy, so moving on, no explanation needed. You all got it right. Okay, this is from the 2013 released exam as well. This covers exactly what we just talked about, but this is a multiple choice question that is looking at phylogeny. So it's giving you data regarding the presence or absence of a trait. So I wanted to show you this because it's not always going to be percentages. Sometimes they're going to give you a trait characteristic table and you're going to have to piece through like, what is this all saying? So we've got species V through Z. We've got traits 1 through 5. And plus means they have that trait, and minus means they haven't. And then you've got 4 phylogenetic trees to choose from. So take a moment. I'll make a new poll. Ooh, you are all rocking it. Really good job. Really, really good job. So if you're thinking about, if you're lost and you're not answering it yet, remember, find the outgroup first. Which one is least similar to everyone else, right? Who's the outsider? And to me, that's species Y, because I look at this and they have none of these traits. They are the most ancestral. They don't have any of these derived characteristics. So I would put Y out as an outgroup. And that takes away 2 of your answer options. I see Y out here as an outgroup for C, and I see Y as an outgroup here for Y or for A. Then you look at your next least similar, right? And you're like, okay, well, look at Z right here. I've got Z that has traits 1 and 5, and then I have X that has traits 1 and 5. So like, that's interesting. Maybe they're a pair. And then we can look at V and W and notice kind of a similar pairing where they both have 1 and 2 and their only difference is 3. And so to me, the correct answer is A. That's what the data is supporting. And a lot of you said that as well. So I'm really proud of you. Okay, last multiple choice question. And then we'll move on to an FRQ. Just let me pull up a poll. Oh, there's no poll? Oh man, I must have not hit set. Did I not hit go? Oh yeah, I did. All right. Anyway, let's go. So, this is one of those individual questions, but it's meaty. There's images, there's some information about the study, and then there are 4 answer options that are quite large. So, I'll read it to you. A group of mice was released into a large field with which no other mice had access. And by the way, test taking strategy, visualize the setup or draw it out, right? So I'm visualizing a group of mice being released into a large field to which no other mice had access. Immediately after the release, a representative sample of the mice were captured, and the fur color was observed and recorded. So they looked gray, brown, black. How many do we have? The mice were then returned. After 20 years, another representative sample was captured, and the fur color was again recorded. So which of the following best explains the change in frequency distribution of fur color phenotypes in the population as shown above? So you look above and you're like, okay, immediately after release, the brown mice, we just had a lot of brown, right? That was the variation was gray, brown, black, high frequency of brown. 20 years later, our gray mice are doing great. Brown mice, not so like medium. Black mice are about the same. So it's wanting you to choose an explanation that best explains the change in frequency. So A, the allele for gray fur is unstable, and over 20 years, most of those alleles mutated to become alleles for black fur. B, the field was composed primarily of light-colored soil and little vegetation, affording gray mice protection from predators. C, sexual selection led to increased mating frequency of black and brown versus gray and brown. And D, the gray mice were hardest to capture and were underrepresented. And so you have to try to choose the best thing, like what makes most sense. When I was learning about things like this, and we're talking about like fur color and phenotypes and why things change over time in general, like why do things change in general? Because of natural selection, right? Like, yes, mutation can happen, but that's like rare for there to be this big of a difference 20 years later. So B is going to be your best option, that it was primarily of colored or light colored soil. And so that's why the gray mice didn't do so well, or gray mice did do well from predators. Okay, we're doing good. We only have 30 minutes left. We're going to jump into science practice 3, your null hypothesis, predicting results, experimental procedures, and proposing new investigations. So I'm going to hop away and someone else is going to take over this FRQ for me so I can stand up and get a little break. Good luck.
Okay, guys, for this FRQ, just take a few minutes to go through it, and you have 10 minutes. And begin.
Actually, because of time, we're only going to give you 5 minutes for this one.
Okay, change of plans. We're doing
Yep,
5
sorry.
minutes. No, you're good.
Okay, so we are hearing from the chat and we want to honor everyone's time. It looks like if you attended the first night of CRAMS, you did go over this. This is so here, when I see people asking questions about like what unit is this from, try not to isolate yourself in your mind into like exactly what unit this is, because this is a combination of units. It's talking about enzymes. But anyway, so Sabita, thank you for hopping on and introducing this FRQ, but I think we'll just skip it because I want to respect everyone's time. We can quickly answer what a null hypothesis is. A null hypothesis is going to be that there is no relationship between the variables. So think like null, meaning no. So there's no relationship. So in the rubric, it said the null hypothesis would be that there'd be no effect on the light emitted. So null hypothesis, look about what are they predicting is true in terms of relationship, and then you would just claim they didn't. And just a little side note, I don't know if your teacher talked about this, but I always like knowing why. Like, why would you make, if you are a scientist and you're putting all this time and money into a whole experiment that are years of your life potentially, why write a hypothesis that is the opposite of what you really believe? But here's what it is. Yeah, the answers are up right now, everyone. In science, we never want to say like, hey, my research study proves that fireflies, blah, 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 right? Like you can't prove anything. You can support something with evidence. And so it's a lot easier to write a null hypothesis that says temperature has no effect on the amount of light emitted. It's a lot easier to reject the null and be like, actually, I have evidence in this experiment that I made that rejects this null. And it supports the fact that the regular hypothesis. All right. So I hope that helps as to like, I remember when I was a student and they were telling us to write null hypotheses. And I was like, why would I write a hypothesis that's the opposite of what I'm trying to show in my experiment? That makes no sense. But again, it's just because in science, it's a lot easier to show support to reject a null rather than not. All right. All right. We got this, everyone. We are in the home stretch. Okay, so this is the 2016 FRQ. And so now I'm a little nervous that I didn't look at all of Mrs. Shoemate's slides because she did like 112 and I had 100 and something too. But this is a 26 FRQ 2016 free response question. And all I want you to do right now is look at the information and try to think about what's the dependent variable, what's the independent variable, and possible controls. This one, you can't necessarily just look at the x-axis for independent, okay? Because time, it's not the independent variable here, okay? So take a moment and just wrap your head around this graph. All right, so in a real test situation, you'd be given more information than this. You'd be given the story of what this is all about. But really what's happening, it says bacterial growth is bacterial population growth in the presence of two nutrients. And so they are measuring bacterial growth. So follow my little red line. Okay, this is the line for bacterial population density. And this is going to be your nutrient 1. And this is your nutrient 2. And that goes here. So there's these two bacterial populations, or this one bacterial population grown in the presence of two nutrients. So they're wondering, how is the bacterial population density going to differ based on those two things? Oopsie, that's not what I wanted. So the dependent variable is what they're measuring. So that's your bacterial density. That's what they're measuring. The independent variable is the nutrients, nutrient 1 or nutrient 2. Which one is going to affect the population density? And then your possible controls. Thankfully, there's so many options for controls. Just think of anything where you're not using treatment on something. So maybe I'd say, hey, we're going to have a bacterial population grown on no nutrient, or bacterial population maybe just grown on nutrient 1 and then grown on nutrient 2 separately rather than together, so that we can kind of see the difference between the two. And then obviously another control would be like using the same bacteria culture, right? Like don't use E. coli and use a totally different one. Okay. I think I'm going to skip this for right now. Yeah, I'm going to skip these. We can always do them on the last night. All right, we have 25 minutes left. Take a deep breath. I'm checking in. I'm going to check in with my teacher host to see how we're doing, see if there's anywhere we need to go back to. Okay, we'll take a deep breath. We're almost there. Oh, someone's asking of the independent variable. Back here, the independent is like I, the experimenter, am doing like what am I putting out there to test? So I would say the nutrient 1 and nutrient 2 would be the independent and dependent. And that's not a question that was asked on that particular FRQ, but I just wanted to kind of have us take a look at that. Okay, we are almost done, like I said, and I do want to give some time for a chance for us to look at the Q&As, and I'll try to on the fly. I will try to on the fly answer some of them. So let's shift to Unit 8, symbiotic relationships. If you don't remember them, that's okay. But I've got a mosquito and a human, and then I've got a clownfish and an anemone, and they are different symbiotic relationship pairs. One is a good, positive relationship, and one is not so much. Okay. Does anybody remember what type of relationship a mosquito and human have? So think about what type of relationship do a mosquito and a human have? Hopefully you said parasite. And then the other one is mutualism. So the clownfish benefits, and so does the anemone. So the reason I wanted to talk to you about that is that this next FRQ is about symbiotic relationships, and I thought it would be easier to look at. So I'm going to move myself out of the chat just so that you have more space to read. Take a look at this one, and you don't have to necessarily graph it, but try to visualize how you would answer this question, okay? There you go. All right, so this one is showing you the graph above shows the mass of plants from two different species over time. So we've got species 2, the dotted line, species 1, the straight line. The plants grew while attached to each other. Oh, sorry, I cut that off. The plants were separated at that time, indicated by the vertical line. So here they're separated. Using template 1, graph the predicted shape of the plant mass lines after separations if the plants were in an obligate mutualistic relationship. Now, if you don't know what obligate mutualistic is, you should know what mutualism is. And you should know that, like, hey, these two things really rely on each other if they have a mutualistic relationship, right? Like the clownfish and the anemone. So if they're really relying on each other and then they're separated, what might those upward curves start to do? And then conversely, template 2, they want you to graph the projected shape of the plant mass lines if species 2 was a parasite of species 1. So these are all just hypothetical things, and you just have to think through, okay, well, they both were going up, but if it's a parasite, what's going to happen to the two? So I'm going to give you 30 seconds to think about that. How would you draw those lines? Now that you've had some time to think about it, I'll show you what the accepted answers were. You wouldn't have had to draw a shaded in line. This is just representing the range of lines that they would accept. Okay, so they both are showing a downward trend because they were doing well because they relied on each other. And if you take them away, they're going to do worse. And then in a different way, the parasitism one, the parasite's not going to do so well if its host is gone. And if the host doesn't have a parasite anymore, it's going to do well. And so that's why we see this line going up and this line going down. So again, this is just the information was given there to you, right? You really just needed to know the difference between mutualism and for parasitism, okay? All right, we're doing great. I'm going to skip the big information. So what I have planned for the rest of today was to kind of look at some like real data and then go through, you know, pulling out their hypotheses, pulling out their claims. But I would like to jump into just some practice problems instead. I would, because I feel like I want to honor your energy levels and not overwhelm you with something that's like, you know, from a real published research study. So I would like to take a vote as a group. And so I'm going to put a poll in so that you can all have a voice in how our last, 15 minutes goes. So I'm just going to make a new poll and I'd love for all of you to respond. How should we spend our last 15 minutes? All right, I have created the poll. I hope you can see all the answer options. So how should we spend our last 15 minutes? Where did it go? Did it go to the top? Oh, yeah. That's weird. Okay, so the poll is like in the mid is the second question. I don't know why it's not showing up at the top. So how should we spend our last 15 minutes? Its options are practice quizzes with the music, another FRQ, a few multiple choice questions, or look at the most voted poll questions that you've all been asking and then answer those. Okay, so your voice, you tell us what you'd like to do. Okay. It looks like, oh man, we're split. Darn it. Okay, so 38% of you said practice quizzes with music, 43% said another FRQ. So we've got time to do both. So let's just do an well, let's do some practice quizzes first, just to kind of mix it up and get our brains doing something else. And then we will do an FRQ to end the night. And then we've also got some reminders about a survey to do. So give me a moment here. All right, let's do Unit 8 because we didn't get to ecology practice questions very far yesterday. So just give me a moment. Thank you all. We're in the home stretch and you're all doing so well. I just want to remind you, make sure you're staying PG in the chat, that, we're here to be respectful to each other and not be like ultra silly and immature, okay? You're all wonderful people and I know that you can do this. Okay. Hopefully you can see my screen and then I'm going to put the quiz. I'm going to wait, actually, here we go. I'm going to put this in the chat. So look for it in the chat so that you join our quiz, okay? I just put the link in a moment ago. Put it in one more time. Okay, now I'm being annoying about it. Put it in twice. All right, so we left off on this question the other night. A group of same species living in the same area at the same time. That's going to be your population. So population of organisms, same species, same area, same time. Boom. Number 5. Okay, so N is your population size. That is something that will be given to you on the equation sheet. All right, so don't worry about, oh no, I don't know all those letters. It's on the equation sheet. All righty. Good job. All right, so a graph that seems to experience exponential growth and then plateaus is going to be your logistic. The reason I know some of you wrote J curve. J curve is how we describe logistic growth, or no, sorry, J curve is describing exponential growth. The S curve is describing logistics. So just be careful with that when you, kind of similar to what y'all were saying earlier about like, oh, can I just talk about enzymes like lock and key? Like you can't. Those are silly ways for you to remember what an enzyme does, but you need to show an AP reader that you know what that logistic is the exponential growth. Alrighty, next question. Let's go. Excellent job, 92% predation. Woo! Wonderful. It's density dependent, meaning if these organisms are all living really close together, then this factor is going to influence them more, right? So like if you're organisms that aren't spread out, predation isn't going to affect you as much. Whereas if you're in a big clump together, then that could be a limiting factor to your population size. Next question. Amazing. Higher death rate than birth rate. Perfect application to the population size calculations. Good job. Oh no, the song ended. All right. Keystone species, excellent. We talked about that last night. They are super important. They're the keystone. If they disappear, the whole entire ecosystem could collapse. All right, this will be our last multiple choice question. Get your answer in. Wonderful. All of the above. So I have good news and bad news. The bad news is it's very unlikely that you will see hold on, let me put myself back. It's very unlikely that you will see all of the above answer on an AP test, right? Like that would be pretty unlikely. But the good news is I want to show you something, and then we'll do one more quick FRQ. Hold on. And by the way, you have access to these slides. I'm trying to find the survey thing, Becky. Where is it? I skip it. All right, everybody. I'm trying to show you the good news, which is a survey for you to take. And I cannot find it. So 1 second. Okay, I'm at the end of my slide presentation, so I don't see any survey. I don't know what's going on. Becky, can you help? I don't know where the slide is with the survey. It's linked. Okay. Okay. Well, by the way, it's important for you. Oh, here it is. It's right here. AP Bio Night 3 survey. All right. It's been a long night. It's been 3 hours, everyone. As you know, it's been a long, long night. Okay. So let's just go for one more FRQ and then we can call it a night. But I do want and ask you to, I want to ask you to take that survey. It's really helpful. All of your information that you could give us will help us do our job better. And I really appreciate that. So here's your FRQ. This is from a 2013 released exam when they used to have 6 short FRQs instead of 4. So this one is a very short FRQ. So I'm going to give you 3 minutes to think about this one. This is from the 2013 released exam. All right, I think that was 3 minutes. I didn't put a timer on, my bad. So this is saying matter continuously cycles through an ecosystem. A simplified carbon cycle is depicted below. So here's the simplified carbon cycle. It's showing CO2, something happens, and it's an organic molecule. Over here, we have organic molecules, something happens, and we've got CO2. So A says identify the key metabolic process for step 1 and the key metabolic process for step 2. So even though there's only one bolded word there, they're looking for a process for step 1 and a process for step 2. Then briefly explain how each process promotes movement of carbon. And then for each process, your explanation should focus on the role of energy in the movement of carbon. So this does not mean, give every single step of cellular respiration and photosynthesis. That would be wild, and that would be not what they're looking for. This is about what you'd be looking for. So here's the identification. Step 1, you could have said photosynthesis or Calvin cycle. Because remember, go back, step 1, we've got CO2 made into organic molecules. So yeah, that makes sense. Plants use CO2 and they make it into an organic molecule. And then step 2 would be cellular respiration, where we're taking an organic molecule and CO2 is released. It's very simplified, right? The explanation, I know this is kind of weird to look at. This is like for an AP grader to look at. And so your explanation for photosynthesis would be its role of carbon, right? You'd be like talking about CO2 is fixed. The role of energy is that it uses light energy, and then the output is organic molecules. And for cellular respiration, organic molecules are or you could just say broken down, that's fine. And the role of energy is that it's using it in ATP production, and then the carbon output is CO2. So each row, you get 1 point for each row. So if you said something along those lines, you would have gotten the point. Oopsie. Okay. Yes, you've all been sitting here so well. I can't believe it. All right. For the explanation, if we go back to what the prompt is asking, remember, answer the prompt. It says we've explained how each process promotes the movement of carbon. And so you'd say, okay, well, carbon is being fixed and made into an organic molecule. And then organic molecules are being broken down to make ATP. That would be like how it promotes the movement. And then also the explanation should be the role of energy. And so you wouldn't necessarily need to get into super in-depth explanations. Okay. Yes. I see some of you are talking about sending snaps. Don't do that here. Thank you. All righty. We've got 2 minutes left. So I'm going to ask one or more of my student hosts to come on real quick and just give you one last word of encouragement or word of advice from their experiences. Hopefully they'll see you tomorrow night. So any of my student hosts, you don't have to show your face. You could just give a verbal, but come on up here and just give a word of advice. Thank you. Oops, I want to hide myself. Sorry. There we go.
Okay.
I'll start.
Oh,
Oh, sorry, you can go ahead.
so I took the AP Bio exam 2 years ago, and I just wanted to say that no matter how confused you might feel right now, or how stressed out you might feel, at the end of the day, this exam is just that. It's one exam, and no matter what score you achieve, it does not tell you anything about yourself. It does not define any part of yourself. And by the end of May 10th, or by the morning of May 11th, I can promise you guys that the majority of you guys will be like, wow, I can't believe I worked this entire year off just for like a simple 3 to 4 hour long exam. And that's it. Like, that's it. And here at Fiveable, we fully believe in you guys. I mean, the fact that you guys are showing up for 3 hours, 5 days in a row, sometimes at like, some of you guys, it's your 2 a.m. That tells you so much about how hardworking and how much you guys push yourself, which is a much better metric to measure yourself by than the score that you achieve on one exam. We fully believe in you guys, and we know that you guys are going to do great, no matter what score you get, because you guys put in the effort, and that's what really matters. So thank you guys so much for joining.
Yeah, and adding on to that, you guys literally did so much more just by like joining the session and staying for 3 hours. So you guys got the studying part down, obviously. And you have to remember, you have to put yourself first before any exam because the test, it just comes and goes and you're just going to be there forever.
Honestly, Rashi and Samita did a very good job with what I was gonna say. So I'll leave a little bit like a comment for words of encouragement, just so I'm not rambling for too long. But you guys did amazing, and us at 5 are so proud of you guys.
You're muted.
I'm that old person who doesn't realize that I'm muted. Thank you. Oh man. All right. Well, what I was saying is that it's 10 o'clock. And so our session is over, but I love the words of encouragement that our student host gave because it's completely accurate and you all showed up and I appreciate it. Loved getting to interact with you all and hopefully you jump and, join in for the two next nights to get you ready. Have a great night, everybody. Bye.